In this video, we will discuss about the expression of FAS-DNR enhances anti-apoptotic role and the in vivo persistence. And this time they used different cancer models in the to infect the mice. So first, what they did is they transduced this T cells. They co-transduced the T cells with the empty vector and the CD19 CAR, and also they co-transduced the T cells with the CD19 CAR and FAS truncated intercellular and FAS truncated region. And what they are able to see is that in both the cases they are able to see uh, almost equivalent number of transductions that can be seen in this panel B, where we can see that the amount of Thi 1.1 positive and alpha CD9 positive CAR cells are same in the first condition and also in the second conditions where FAS region is truncated. Thereafter, they check the effect of this CAR when they co-culture with different cytokines or media. So first they co-culture them with FASL ligand and they are able to see some death in the empty vector construct. But when they use the media, they see a slight change or slight decrease in the media compared to the uh, slight decrease in the case where empty vector is transduced along with CD19 compared to the case where CD19 is transduced along with FAS has been truncated. But when they use this anti-CD3 and anti-CD28, they are able to see more of the death in the case where empty vector is transduced into the cells, because it might trigger FAS signalings that will trigger apoptosis. But there is less, almost negligible death observed in the case of where FAS intercellular FAS signalings has been truncated. And when they use these tumor cells, that is E2A PBX, they are able to see some death in the T cells that has been transduced with anti-CD19 CAR and empty vector compared to the case where T cells are transduced with anti-CD19 and FAS intercellular part has been truncated. So once they find out that the region is the FAS signaling is trigger apoptosis in the case where its intracellular parts is not been truncated. So they wanted to do this studies in the in vivo also. So they took the mice, inject them with this virus tumor such as E2APBX, that is PDBAL, and then after 5 days they inject this virus. After 5 days they inject the virus, and they take the mice, and after 5 days they inject the virus, and 1 day after that they infuse this co-transduced anti-CD19 CAR empty or FAS truncated region, and for 2 days they also inject IL-2 to expand the amount of these T cells, and then measure the this CAR T cells, transduced CAR T cells after 14 days in the spleen and bone marrow, because these are the two places where this kind of tumors are being accumulated. And they are able to see that the T cells are almost negligible in the case where empty vector is used at both spleen and bone marrow compared to the case where FAS intercellular part has been truncated. And thereafter they also check the amount of these tumor cells, and they can see that tumor cells are around 70% in the case of PBS and also in the case of empty vector. But in the case of this FAS intercellular part truncated region, they see almost negligible tumor cells. So this indicates that this by inhibiting this apoptosis or by using the FAS-DNR can enhance anti-apoptotic factors that might help the T cells to work on the tumor cells. And thereafter they also check the survival rates at different concentration of the CAR T cells transduced. So when this 3 × 10⁵ cells are transduced, they are able to see the mice with the empty CAR construct can survive up to 55 days, but with the mice with the FAS signalings truncated can survive for a longer time, and around 100% of those mice has been able to survive. but when they transduce the cells with 2 × 10⁵, they see the survival rate for the case where the empty vectors goes down, whereas there is no change in the mice that are transduced with CAR with FAS has been truncated. So this and they also mentioned that they are not able to check whether 41BB pathway has been triggered in this case or not, but the difference is clear from the results that when the intracellular FAS signaling has been truncated, it will enhance the efficiencies of the CAR, and that might result in the increased survival rate. That's what they have. That's all they have shown in these results. I hope you understood their effect of this FAS-DNR, that is dominant negative receptors on the anti-apoptotic role and its in vivo persistence. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.